Great. Well, thank you so much for having me. My name is Leila Zegna, as Jill said. I'm the mother of Roya Zegna, our beautiful little 6-year-old girl who has Kabuki syndrome. And I'm also a board member of KSF and the co-founder alongside my husband, Eduardo, of the Roya Kabuki program at Boston Children's Hospital. So in this session, we're going to be talking about the clinical trial pipeline, and that's an incredibly exciting area of research and development. We heard through countless interactions with other patient families, through our clinicians, and through the surveys that have been conducted over the years, that what matters to parents and patients alike, above all else, is the neurological aspects of Kabuki syndrome. In our 2021 crowdsourcing survey, parents indicated that the neurodevelopmental and behavioral symptoms was the area that most heavily impacted individuals with Kabuki syndrome. So out of about 106 participants in that survey, 84% indicated that they would like to see more research and therapies that could improve cognition, including areas like thinking, memory, and learning. Therefore, that has been the key focus for the therapeutic development and the clinical trial effort, focusing on the neurocognitive and developmental profile of Kabuki syndrome. So in this session, we'll hear from a really outstanding group of drug developers, researchers, and clinicians about the opportunities ahead to take a compound or a set of compounds through a clinical trial for Kabuki syndrome. We'll learn more about the upcoming clinical trial run by Oryzon Therapeutics, as many of you have had questions in the previous sessions about that, as well as the importance around establishing the infrastructure to ensure that trials are set up for maximum success. And so that will include a deeper dive on how to develop and design consistent outcome measures that are specific to Kabuki syndrome and how to measure a therapy's success. As we've talked about in previous sessions, a standardized scale for Kabuki syndrome would really enable researchers and physicians to accurately assess the severity of symptoms both before and after treatments. And it would substantially improve the likelihood of success and accuracy in any clinical trial going forward.
So, I'm going to kind of fly through because I know we've lost some time now. One thing we have talked about, Dr. Bjornson talked about this earlier, and some of the other presenters, is that one thing that's very interesting about Kabuki syndrome as an epigenetic disorder is that there may be ways to perform therapies that specifically treat the underlying symptoms of Kabuki syndrome without necessarily correcting the broken KMT2D or KDM6A protein or gene. And this is the idea of restoring the balance between open and closed chromatin with an epigenetic modifier. And so the goal of this would be to create clinical trials. And in order to create clinical trials, which clinical trials are, as you heard Dr. Hartman talk about earlier, what you absolutely need in order to get these therapies to be able to be delivered to patients. And a well-designed clinical trial has a number of features. It has a specific patient population. Yay, we get a check mark on that one. Okay, you all are here. It has a therapy or intervention being tested. That's what you've heard a lot about from different aspects today. It has knowledgeable and collaborative teams, which I think and hope that we being the scientists, but also you all. And then it has appropriate outcome measures. And that last one is where I focus a lot of my work. And so what is an outcome measure? So an outcome measure is how we measure whether or not something actually makes a difference in a clinical trial. So we're measuring the symptom or the underlying disease mechanism of interest to see whether or not it changed across the course of a trial. And in order to do that, it has to have a few features. So it has to be reliable. And what that means is if I test a patient on day A and a patient 3 days later without any intervening change, I want that to be the same. Because if it's not the same, then I don't know whether or not the change later on was due to the therapy or was just inherently a bad measure. It was not a well-chosen outcome measure. It has to be valid and clinically relevant. And what I mean here is that I could easily measure with precision accuracy how many ankle sprains someone with Kabuki syndrome gets across the course of a 6-month trial like the HOPE trial. But at the end of the day, that's really not valid or relevant. Yes, it's measured and it's, I can count it exactly, but who cares, right? And so it has to actually target the symptoms that are of interest. And then there has to be variation, meaning whatever measure you use, that has to be able to change over time. Otherwise, we can't see whether or not the therapy changed it. And so as Dr. Bjornson touched on, the Kabuki mice that his lab developed have these hippocampal memory defects. And this is shown by the Morris water maze, which again, he showed you this data a little bit before. And it turns out that this matches with what we see in the humans. So this is a study we published a few years ago that shows that compared to individuals with the same IQ who don't have Kabuki syndrome, so these are controls matched by IQ, that people with Kabuki syndrome do the same or even a little bit better in some areas like their full scale IQ or their verbal and language measures, but specifically in the visuospatial areas, they're universally doing much, much worse. Those are the red bars that are clustered over at the end. And so this lets us know that this is a symptom that we really want to target in Kabuki syndrome. And as such, we have designed outcome measures that are used in clinical trials, including the diet trial that we're running out of Kennedy Krieger and planning on expanding to younger ages and more centers, and the HOPE trial, which is what today's segment is about. And then the therapy or intervention being tested. So again, we have a couple options that I'm going to talk about today. So you heard a little bit about this from Kristen and Matthew. They both came to Kennedy Krieger to participate in this trial. And we know that again from Dr. Bjornson's lab, in the mouse models of Kabuki syndrome, that they get better on a ketogenic diet. And we know from some other aspects of that paper that the reason for that is because of something that's produced when you're on a ketogenic diet that's called beta-hydroxybutyrate. Well, there's this other therapy that's been used in other neurologic conditions, particularly seizures, called a modified Atkins diet that can also produce beta-hydroxybutyrate and also have neurologic effects. And so we're doing this pilot clinical trial in adults with Kabuki syndrome, looking at this therapy to try and treat the cognitive and behavioral symptoms of Kabuki syndrome. And you're going to hear more in a little bit about all the ways that we test, but the outcome measures that we focus on are based on these visuospatial functions, as well as some other things that we'll talk more about. So the next steps are to expand this to school-age children. We may add other centers, and you're gonna hear more about the cognitive and anxiety outcome measures with biomarkers. And you all participated in this crowdsourcing survey that says you like the idea of exogenous ketones as well to get this beta-hydroxybutyrate level up, and we may include those. So now we're going to move to what we're talking about in the HOPE trial. So again, you saw some of this data from Dr. Bjornson. This is all from his lab. The LSD1 inhibition, LSD1 is also known as KDM1A, but it removes the methyl marks at the same place that KMT2D is supposed to add them. And as such, when you lose KMT2D because you have Kabuki syndrome, or you lose some of the function of it, then it makes LSD1, who has normal function, kind of overactive from a balance perspective. And so the thought is, if you can block this a little bit, you can block this eraser, this remover, then perhaps you can restore that balance that we've been talking about. And so again, this is not with this is not using Vafidemstat from Oryzon, but a different LSD1 inhibitor, but the concept is the same. And Dr. Bjornson's lab shows that it does, in fact, modify histone methylation levels in mouse hippocampus, which is what the goal was. So that's very nice. But more importantly, it also rescues the defect of adult neurogenesis in the dentate gyrus, which is that visuospatial area, which is, again, getting back to the specificity of outcome measures. And it rescues those hippocampal memory deficits in the mice. So the mice look more like their wild type counterparts as far as their performance on those Morris Water maze tasks. And so that is just a brief overview of how everything works together to, sort of, we got to use the right outcome measures, which we get from the science and from you all, and we got to use the right therapy, which we get from the science and from our industry partners like Oryzon. And so to tell you more about the HOPE trial specifically, I would like to introduce Michael Ropacki, who is the Chief Medical Officer of the CNS division of Oryzon. And Michael, I'll let you take it from here.
All right, thank you. Can everybody hear me all right?
Yes.
That is a yes. All right, perfect. I'm going to go ahead and share my screen. And please confirm that this is the full-size version.
Yeah.
Okay, perfect. All right, so I'm Michael Ropacki. I'm the Chief Medical Officer of CNS Clinical Development at Oryzon, and I'm going to give you an update on HOPE, which is a double-blind, randomized, placebo-controlled, multi-center phase 1/2 trial to evaluate the efficacy and safety of Vafidemstat in subjects with Kabuki syndrome. Thank you for the opportunity to be here and to present to you today. These are the disclosures that Douglas briefly showed earlier. We have to present these for legal reasons. Just says that it's a publicly traded company. Okay, so moving on to HOPE. It's an adaptive randomized double-blind phase 1/2 trial with Vafidemstat and Kabuki syndrome type 1 patients. So what does this mean? So phase 1, we're going to talk about a little bit more on the next slide, but what this means is that it's got two parts to it. And the first part is phase 1, where we're going to try to evaluate safety and tolerability, and we're going to try to determine what is the right dose for adolescents that have Kabuki syndrome type 1. The second part is phase 2, and this is to look at how well does the drug work at this dose that we found in phase 1 in the adolescents and at 1.2 milligrams for adult patients. We filed what's called an IND, or we're filing what's called an IND, which is it's a application, if you will, to the Food and Drug Administration to allow us to do this trial. And we're hoping that we will get our approval and be able to begin in 2023, and that this could set the basis for expedited development in the Kabuki syndrome, those with Kabuki syndrome. Now, before I go on to the next slide, I just want to tell you that this is an update. So last time I was here, I told you how we'd like to do a trial and what we were thinking about in doing a trial. And thanks to a whole bunch of individuals, that some of them that are presenting at this meeting, like Dr. Jackie Harris, we've made significant progress and hope to be able to start actually enrolling subjects next year. So continuing on these updates, we developed what's called a hybrid decentralized clinical trial protocol. So what does that mean? What that means is that this is only going to require that patients and or their caregivers and family members only have to go into the office 3 times over about a half a year trial. All the other visits will be done remotely, meaning that they will either come to you or you will go somewhere nearby and have all your data collected. The trial involvement, as I said, is about 6 months. The visits at the beginning are a little more frequent to make sure that the drug can be safely administered and tolerated by those taking it. And at the end, it'll be a little bit more spread out because we want to make sure we have enough time to show that we can change things such as cognition. Now, the two different parts of the trial that I alluded to earlier, the first part is to establish safety and efficacy. So how well does it work and how safe is it in patients with Kabuki syndrome, and those 18 to 25. And the second part is that we're finding the right dose. Actually, those are reversed, so I apologize. All procedures for adolescents and the adult patients are identical, and the time to be involved in this trial is the same for both groups. So other things that have been done since the last time we talked, we've identified our sites. We're in the process of getting these sites on board. There is a possibility this may become a global trial, meaning we'll not only do it in the United States, but we may do it in some other countries around the world. Regardless, Dr. Jackie Harris is going to serve as the PI of the HOPE trial, and we will collaborate with other sites and other doctors that specialize in treating patients with Kabuki syndrome. Those that get in the trial will all have to have a definite diagnosis of Kabuki syndrome per the International Consensus Diagnostic Criteria, and genetically confirmed loss of function mutation in the KMT2D gene. Overall, the goals for HOPE are to establish that Vafidemstat is safe and to figure out what the right dose is, and to determine if it improves cognitive abilities, mood, focus mainly on anxiety, and global functioning, as well as to establish the profile of safety in long-term treatment. As I said before, so all collaborators and companies have been identified and contracted. Discussions are ongoing with the FDA. The application with the FDA is going to be filed before the end of the year, and we're hoping to start in 2023. And with that, I thank you for your time and attention, and I will turn it back over to whomever, if Douglas is coming back on or if we're going straight to questions. Thank you.
I can try to come back on, Michael. Thank you. If you stop sharing, I can see if I can share. I've changed computers. And
Did it stop?
yes, it did. So I'm hoping that my slides will progress.
That looks
Can
great.
you verify? Okay. I will skip through, in the interest of time, some of the early things I said, of course, and focus on Kabuki syndrome. We talked about DNA being our destiny. I know you couldn't see slides for this, but I believe you understood what I was saying. And the important thing that we've recognized more recently is that all the information is in the DNA, but other factors affect the way DNA is read, and things like the environment, things like history, food intake, all of these can work with genetics. This is epigenetics to influence, what we look like, how we behave, our phenotype. And our goal is to use epigenetics to try to help to mollify or correct some genetic disorders. For example, in the DNA in some of the people on this very call with Kabuki syndrome, there's a mutation in the DNA. One of the genes, MLL2, as you've heard talked about before, is not expressed. And the result of that is Kabuki syndrome. And our hope is that by using epigenetics, which doesn't change the DNA, but changes the way in which the DNA is read, that we can help people with Kabuki syndrome overcome some of the things that bother them, some of the things that affect their life. We've talked about DNA before, we've talked about chromatin. I just want to give an astounding, what is always astounding to me, piece of biological information. Every cell in our body contains DNA, as you know, but if you took the piece of DNA in every cell in our body and you stretched it out, it would be 3 feet long. 3 feet, as long as your arms spread apart. How is all of that DNA packed into every cell in our body? It's wrapped around a group of proteins called histones. And these proteins form sort of bead-like structures, beads on a chain. This is packed into something called chromatin, and the chromatin is packed into chromosomes. So all of this DNA is tightly, tightly compacted. The way in which the DNA is read is modified by writers, like the gene we just talked about, the KMT2, and also by erasers, enzymes like LSD1. And what if we could, the goal of epigenetic therapies is to use a medicine to either affect the writer or affect the eraser, and by that way, change the way in which DNA is read to benefit people. Another way of looking at this is that a lot of DNA is tightly compacted. Other areas of DNA are actually read relatively easily. And through epigenetic therapies, you can make hard to read DNA easy to read, and so new genes are expressed, or easy to read DNA hard to read, make it difficult to express, to turn off genes we don't want expressed. LSD1 is a critical enzyme, a critical gene that's involved in many parts of central nervous system function. It's the most common epigenetic enzyme in the brain. It's very important for the growth of nerve cells and brain development. It controls certain genes that cause central nervous system disorders. And imbalances in LSD1 or in the writers, like DMT2, causes differences in methylation and methylation balance. And I'll show you a cartoon of that in a second. But perhaps we believe by using LSD1 inhibition, we can correct that. And our drug, has been mentioned several times now, Vafidemstat, is an inhibitor of LSD1, which is specifically used for central nervous system disorders. Its mechanism of action we discussed, but the point of this is that there's a balance in epigenetic modulation of DNA function. There are writers that, like MLL2, that cause the balance to shift this way, and other erasers like LSD1 that cause the balance to shift this way. When we're missing one of the writers, the balance shifts heavily in direction there's no more balance. But if we inhibit LSD1, we believe we can get the functional epigenetic status of very important genes in the brain to be in balance again. I'll mention, I don't know if Michael mentioned this, I don't recall, but we've had over 300 people who've been treated with Vafidemstat, including normal volunteers. It's been, to our great satisfaction, very safe and has demonstrated effectiveness in a number of other diseases in small trials. We've talked extensively about the model of Kabuki syndrome that Dr. Jornsen developed, and Dr. Harris showed in this model that using an LSD1 inhibitor improves cognition, memory, anxiety, and immune disorders. So we're very excited about the possibility of using Vafidemstat to help people with Kabuki syndrome overcome some of the burdens that they face. And importantly, there are a whole family of diseases related to Kabuki syndrome, other losses, genetic losses of DNA writers or DNA readers that we believe LSD1 inhibitor Vafidemstat might be able to help. Kabuki syndrome will be the first demonstration that we can provide benefit to people. And lastly, we've talked about LSD1. As I mentioned, we're developing it for common diseases, common neurodevelopmental or neuropsychiatric diseases. But more importantly, we think that its best use may be in personalized medicine in rare diseases like Kabuki syndrome, where the pathogenesis is very clear, the molecular diagnosis can be made. And this allows a smart drug to be designed that focuses specifically on what it would take to re-achieve balance of DNA expression in the brain. As I said, we hope that it'll be possible to extend this even beyond Kabuki syndrome to help other people with genetic diseases that affect the central nervous system. So let me stop there and thank you very much.
Thank you so much, Doug, and I'd now like to introduce Allison Kalinousky. Allison is a human genetics PhD candidate who works in my lab and Dr. Bjornson's lab, and is going to talk to you today more about biomarkers and outcome measures for trials in Kabuki syndrome. Allison?
Thank you for that introduction, and thank you for the opportunity to speak today. I'm going to be talking about our group's work at identifying biomarkers and designing outcome measures for clinical trials in Kabuki syndrome. So far, we've seen promising results when testing different treatments using mouse models of Kabuki syndrome. So the theory behind these treatments is that individuals with Kabuki syndrome are thought to have an imbalance between open and closed chromatin. And so therapeutics can potentially restore this balance. So some examples of these treatments are HDAC inhibitors, which prevent the removal of acetyl groups on your DNA. So your DNA is folded to fit into a cell. So where the acetyl groups are tells the DNA where to be more open, which allows the genes in that region to be expressed. So an HDAC inhibitor can therefore promote open chromatin and help restore this balance. A ketogenic diet has a very similar thought process. So when your body's in ketosis, you naturally produce an HDAC inhibitor called beta-hydroxybutyrate, which is a ketone body. Beta-hydroxybutyrate can cross the blood-brain barrier and has a similar mechanism as a medication form of an HDAC inhibitor. And then lastly is an LSD1 inhibitor, which Dr. Harris had spoke to you guys about. So using all of these treatments, mouse models of Kabuki syndrome have improved neurogenesis, or the growth of new neurons, and improved cognitive performance. So the next steps is to translate these treatments to clinic through the use of clinical trials. So for rare neurological diseases, if you do everything correctly, there is an 18% chance of a clinical trial being successful. However, if you utilize biomarkers, this success increases the success probability by 40%. So this is greater than the initial success probability. So what is a biomarker? A biomarker is defined as an objective, quantifiable characteristics of biological processes observed from outside of the patient. So biomarkers can be measured accurately and can be measured over time. So you can measure a biomarker before and after treatment to quantify a response to therapeutic intervention. Some examples of biomarkers in other disorders is, say, blood pressure for stroke risks. You can measure blood pressure and use this result to correlate to an individual's risk for a stroke. Also, MRIs are used for Alzheimer's disease. You could look at brain shrinkage and correlate that with the outcome of cognition. So overall, the thought of the biomarker is to look at and measure at a single point in time, and then you can use that to correlate with a functional outcome. So to identify biomarkers, we're performing MRIs looking specifically at the hippocampus. So the rationale behind this is that previous work done by the lab showed decreased growth of new neurons in mouse models of Kabuki syndrome. The mice also performed worse on memory-driven tests. So this makes sense because the growth of new neurons is thought to play a role in memory and learning systems. So our goal of the MRI is to validate that the deficits we see in the mouse models are truly representative of patients with Kabuki syndrome. We then will compare the hippocampus of individuals with Kabuki syndrome to age and sex-matched controls, where if we see a difference, this could potentially be a biomarker that is specific to Kabuki syndrome. Another potential biomarker is an ERP, which we use to approximate a phenomenon called long-term potentiation, or LTP. So what is LTP? LTP is when the connections between neurons become stronger with frequent activation. It's thought to be a way in which the brain changes in response to an experience, meaning it may be a mechanism underlying learning and memory. We think that people with Kabuki syndrome have differences in learning in LTP, which can make ERP a good biomarker in future clinical trials. So it's been shown in mouse models of Rubinstein-Taube, which is another Mendelian disorder of the epigenetic machinery, that they have deficits in LTP, or this difference in learning. As there are many similarities between Rubinstein-Taube and Kabuki syndrome, We are actively testing now to see if the mouse models of Kabuki syndrome also have this deficit. So since we cannot measure LTP in humans the way we do in mice, since we don't have direct access to the human brain in a living person, we are using event-related potentials, or ERPs, to approximate LTP. So for the ERP, it's very simple. We just place a cap with electrodes on the participant's head, then record their brain's response to sound stimulation. We believe ERPs would be a good biomarker because they're easy, tolerable, and inexpensive, meaning it could be done easily before and after treatment. We're also performing ERPs on age-matched controls to look for differences in the way individuals with Kabuki syndrome compared to unaffected individuals learn. Once we define a baseline response, the next step is to see if ERPs can change over time with treatment. And then we can then correlate these results of the ERPs to other cognitive outcomes. So just to switch gears a little bit and to go to a different biomarker, we know that those with Kabuki syndrome have a distinct methylation profile, which is a pattern of methyl groups on DNA that controls which genes are expressed. So as you can see in this picture here, these red dots indicate methyl groups, and where the methyl groups are, it makes the DNA condense, and then things can't bind. So those genes are silenced and not expressed. However, when the methyl groups are not present, the DNA is more open, things can bind, and then those genes are active. So the reason that this is important to know is because both KMT2D and KDM6A are known to affect an individual's DNA methylation profile. And what's interesting is that this methylation profile can be used to diagnose Kabuki syndrome instead of having to sequence someone's genes and look at the changes. So what we don't know is if this signature or pattern correlates with cognitive function and whether the signature is changeable with treatment. So this is what we're actively looking at now. So to do that, we collect blood from participants in order to extract the DNA and run a methylation array, which is a way to look at this methylation profile. And we can collect this before and after treatment. We could also perform cognitive testing before and after treatment, and then we could see whether this changes in their cognitive performance also correlates with changes in the methylation profile. Potentially by restoring this balance between open and closed chromatin that's seen in Kabuki syndrome, we can improve some of the symptoms. And then methylation profiles can therefore potentially serve as a biomarker, and cognitive performance can serve as an outcome measure. So another potential outcome measure is anxiety. So in a recent study done by the lab, we administered a set of behavioral assessments. And while individuals with Kabuki syndrome have very few behavioral problems, we found that 22% of children and 60% of adults with Kabuki syndrome scored over the threshold for clinically significant anxiety. And when you include those who scored below the threshold, but who are actively receiving medication treatment for anxiety, these percentages increased to nearly 40% of children and 80% of adults. And this is significantly higher than we would expect to see in the general population, and also higher than we would expect to see for individuals with intellectual disability. And in reality, these numbers may even be an underrepresentation because the assessments we use can miss a lot of symptoms. We also compared children with Kabuki syndrome to their unaffected biological siblings to take into account any familial risk factors or any environmental factors that can contribute to anxiety, and found that children with Kabuki syndrome have significantly higher anxiety scores than their unaffected biological siblings. So we think it has something to do with the Kabuki syndrome that's causing their anxiety rather than other factors. So knowing about these elevated levels of anxiety matters both for clinical purposes, so clinicians know to heavily screen for anxiety in all individuals with Kabuki syndrome, but it also matters for clinical trials. So it makes biological sense that individuals with Kabuki syndrome would have anxiety, since anxiety is known to be associated with decreased hippocampal neurogenesis. So we can administer assessments looking at anxiety before and after treatment. And potentially, if we see improved anxiety, this can be taken as a proxy for increased neurogenesis. And then, so based on these, the study that we did, anxiety is going to be used as an outcome measure in clinical trials for Kabuki syndrome, such as in the HOPE trial and the expanded diet trial. So just to conclude, biomarkers greatly increase the success rate of clinical trials, and we can correlate biomarkers to clinically significant outcomes. So in our case, we've been correlating MRIs, EEG, and DNA methylation and correlating those with the cognitive function and anxiety. So I've listed the contact information from all the members of our study team, as well as the current ongoing research. So we have MRI, EEG, cognitive testing, and DNA methylation for outcome measures. And then we have the modified Atkins diet that we're hoping to expand to school-age children pending grant approval. And we just want to thank everyone who's already participated, and we hope to be hearing from more of you soon. Thank you for your time. And then I just now want to introduce Dr. Adam Vogel, who is the CEO and founder of Redden Lab, the leading experts in speech protocol development, data analyst, and interpretation for clinical trials.
Thanks, Allison. Can you hear me just before you sign off?
What about now?
Adam, we can hear you.
Okay. I never know with those Bluetooth headphones, and I missed the technical check-in today, so I apologize for that. I'm in Dallas, and we went line dancing last night, and that meant that I slept in, so my apologies. I'm, as you can probably tell by my accent, I'm not American, I'm Australian, and I'm usually in Melbourne. There, I'm a professor of speech neuroscience at the university. And as Allison alluded, I also work at Redden Lab, who do lots of speech. What I wanted, sorry, here are my disclosures. It's just people who give me money, and not me personally, who give our research money and our company and some of our collaborators. So the reason we're here today, as in me and my group, are some of the work that we've done looking at Kabuki syndrome and how speech and language can change over the course of the condition. We know that dysarthria, and I'll go through what these terms mean. Dysarthria is present in many individuals with KS. We know that there are some difficulties with oral motor control, phonological speech errors, I'll again describe what that means, and expressive and receptive language changes as well. So that means understanding and also producing language. To provide a bit more context to what that means, in terms of communication, speech, language, and hearing are usually the three domains that we consider. Language being the cognitive or the thinking around how people form what they're trying to say, like I'm trying to do now. And that relates to the words that we use, but also the rules that govern how we formulate our speech. Then there's speech, which is the motor element to communication. That's how we sound. If we think about perception and language, listening, that's the third element to it. You can see there's actually a little bridge joining this phonology element up the top. That usually represents a combination of speech and language, where people are producing the wrong sound and it sounds like they have a speech issue, but it's actually related to a language impairment that sits behind it. So that gives you context to what we're measuring. We're actually focused today in KS on speech. Now, it's not necessarily straightforward assessing speech in complex conditions like Kabuki, and I just wanted to go through some of the considerations that we have when we're building up a battery on how to assess this particular aspect of a condition. So what do we assess? Do we assess motor speech? Do we assess the developmental delays that we see in neurodevelopmental conditions? Do we assess language, or do we assess social communication? And in that context, that's conversations and people's ability to hold onto a conversation when they're speaking to someone else. I think we can do all of those things, but it does tailor the particular battery to what we have. Do we get that information from patient-reported outcomes? And we certainly have some of them available to us. Are they observation or clinician rated outcomes, where someone is going down a checklist and saying what's going on? Or do we have set tasks or semi-structured tasks where we elicit someone's communication and speech? Do we do that in the home? And we've just learned from Michael that some of this trial is actually remotely delivered and some of it's within the clinic, and that has bearing on how you conduct the assessments, whether or not you autonomously delivering them, as in, is it being done by the patient and their family, or is it being done by a trained rater or the clinician themselves? Sounds like this is a mixed trial that you've got planned here, which makes it really interesting. And when do we assess? Do we assess every day? Do we assess, which is a lot, and I wouldn't recommend it, monthly or 6 monthly or yearly? Do we do bursts where we do 3 tests within a week and then take the average of that? Or do we just do them incrementally over time? As we know, the speech and language features in Kabuki vary as a function of disease duration and age of people and other features relating to cognitive impairment. Some of the tasks that we set can be quite challenging, certainly for the younger participants. If we are doing home data collection, how do we consider the hardware that we use? So are we providing provision devices? Like, do I say, okay, everybody's going to use an iPhone, and that means that Oryzon buy an iPhone for every participant, and then they all get a free one? I'm not suggesting that, but it does sound fun to me. Or do we provide a particular hardware configuration for everybody? It does need to be considered, because the microphones that we use within each device vary, and that means the information that we get from the recordings also varies. Do we get people to just have a conversation and then record that information and measure what's going on there? In some contexts that can work, but in other situations where people aren't very chatty, or they are communicating with someone that they're unfamiliar with, that sort of approach can be challenging. The analysis that we do is largely objective. And when I say objective, that means we're not relying on a clinician to make a judgment on something. We're actually taking features of the physical speech signal and analyzing that via acoustic analysis. And then we're also looking at language features relating to the rules that govern language that I mentioned before. And there's a process that you can use there called natural language processing. So that takes out the element of bias, where clinicians are very good at their job, but for a clinical trial, we need to remove the element of bias that comes in with clinical exams. And I think it's really important, and this needs to be spoken about all the time in clinical trials. We need to make sure that the data is as good quality as it possibly can. So in that context, we do lots of training of sites and of participants. We have lots of standardized materials around manuals that need to be read. People don't tend to read them, so we put as many pictures as we can, but also checking the data when it comes in. So for Kabuki, we're thinking that there's a primary focus on speech, but we're going to use tasks that fit along a continuum of complexity, and I'll just describe what that looks like. Complexity being, some of the tasks are very straightforward in that there are just a motor task where we elicit someone saying a sustained vowel, like just saying ah, and from that we can get lots of information about voice quality and also motor timing. Then we can have tasks which are a little more complex in terms of the articulation that's being elicited, something like saying the days of the week or counting, and then moving further along that continuum to tasks like a syllable string. And these are often used in neurological exams, like a pata task, getting people to say pata, 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 pata very quickly and clearly over 10 seconds. And then we also incorporate tasks like describing a picture or telling a story about something that they like. And that gives us more information about the language as well. So we use tasks that fit along this cognitive motor complexity spectrum to give us information about the speech. I wanted to just quickly highlight a study that is running with Dr. Bodemer up at Boston Children's. They've just cutely called this study the TALK study, and we're supporting that study through the protocol design and the analysis. Just to give you an idea how it works, participants will click on a web link that's specific to your username, and then you will sign into the system and you'll click through a various series of tasks, and it'll just take you through those tasks like the ones that I just described. It's really straightforward, and will provide us with a huge amount of information that then helps us, well, I don't know why it says Poland down the bottom there, helps us get a better picture of what's going on in Kabuki. So very brief. Thank you very much for the opportunity to speak, and I'm very happy to introduce Leila, who's going to host the Q&A now.
Great. Thank you so much. That was really fascinating. And we have so many questions that have come through the chat. So clearly a very engaging session for everyone. There are a couple of questions. I'll try and direct them to the right person. One of them is around, we've talked a lot around the visual spatial aspects of Kabuki syndrome. Maybe this is a question for you, Jackie. So, you know, how does that actually manifest in a practical life sense? So are they more clumsy? Does that mean they can't drive when they're older? You know, what are the practical implications that may limit quality of life as it pertains to visual spatial disability?
Yeah, all of those things. So visuospatial is, what we found is that visuospatial is the primary driver of the cognitive disability in Kabuki syndrome, meaning if you improve the visuospatial functioning in Kabuki syndrome, people become much more cognitively like their peers in theory. And so we're looking at all aspects of visuospatial. I saw a question of this in the chat. Visual motor is included in the battery we do. So it's not just perception, it's perception, reasoning, motor, memory, all those aspects. And it has to do with your math skills. It has to do with your ability to read, actually, because we don't read by sounding out every word. We start to recognize sight words and go quickly to be able to read fluently. And if visuospatial wise that's difficult for you, you're not going to be as functional of a reader. It has to do with how you find your way around, how you process information for things like driving, and your handwriting, your ability to write, and really overall your ability to function. And so these are the targeted measures that we think most correlate with the ability to cognitively function in the world. They're not just, little tiny insignificant things that we can measure. And that's why we picked them.
Got it. That's really helpful. I think there was in a similar vein, there's some questions about practical kind of manifestation. And so in layman's terms, and maybe I'll direct this to Dr. Ropacki from Oryzon, but there was a question around, in layman's terms, what is the HOPE trial trying to accomplish? Is it better memory? Is it speeding up mental processing? Kind of linked to what Jackie was just talking about. Can you talk about maybe some of the specific practical things that you hope to accomplish?
Yeah, so if we improve the visual, spatial, and motor coordination, all the things that Jackie just got done talking about, being able to drive from complex, interactive things like driving all the way down to, tasks such as reading, should in theory be improved. Also, we're looking at visual memory, and so not only when you're reading things, sight reading, if you will, to be able to remember better. But also we're looking at overall memory, verbal and visual memory. And then we're also measuring functional abilities in this trial as well. And specifically, we're looking at how does this impact somebody's ability to just perform their routine daily tasks in their life. So by improving the cognitive functioning, the thoughts are that a lot of these downstream activities such as function, your daily life activities, will also improve. Another thing that we're looking at is whether we can have an impact on improving mood. And specifically, we're going to be measuring various ways anxiety in this trial, because the drug itself, Vafidemstat, although a non-anxiolytic drug, meaning it doesn't make you tired, it doesn't have any of the side effects of a lot of other drugs out there. It, in theory, if it works, should help with the anxiety, or at least the parts that are described to me by Dr. Harris around repetition and asking questions repeatedly and these types of things. So we're hopeful that all those things could be positively impacted by the therapy.
Got it. And given that we're talking about the Oryzon therapy and the trial, There was a question around LSD1 inhibitors in general. I think there was a reference to the fact that there are a number of LSD1 inhibitors that are out there, and some data that was shown from a Takeda LSD1 inhibitor. So is one LSD1 inhibitor more likely to be effective than another for Kabuki syndrome, or given that they all kind of target the same mechanism, they all have a chance to be equally effective?
So when you talk about different drugs, and I don't know all the specific of the drugs that are being referenced here, but in general, when you're talking about other drugs, you not only look at the mechanism of how they work, but you also look at how their safety profile is, as well as how their efficacy. So how safe are they to take, and how well do they work? And everyone in theory works a little bit differently. And again, without knowing the specific compounds, what I can say is that what we do know about the Takeda compounds is that they pulled their program, and it appears from what is in the public domain that this may have to be due to something related to safety. And what I can tell you on the Vafidemstat side is that we've had over 300 participants treated. We've had 9 in human clinical trials, 7 that are completed, 2 that are currently ongoing. Some patients have been on therapy for over 18 months continuously, and the drug, knock on wood, has so far been very safe and well tolerated. So we're very hopeful, no pun intended on the trial name, that this safety profile would continue as we move into the Kabuki patients with Kabuki syndrome.
Great. Thank you so much. Jackie, maybe for you, a few questions on the modified Atkins diet trial. One of the questions is, do you think that the trial would show more effectiveness on a younger population rather than adults? And if so, why would that be the case?
Not necessarily, although obviously when you're measuring, cognitive outcomes, right? You may get to a point in adulthood in which it may be more difficult to change those sorts of things. And so, in theory, children should still be quite malleable for that, although the preclinical or mouse data shows that even into adulthood, these things can be altered for Kabuki syndrome for however well, the mice approximates humans, which in many ways so far is good. But the other thing to know is that the current study that's ongoing in adults is very much what we call a pilot study, meaning it is intentionally kind of very short and limited and designed to look at some safety and feasibility issues, and the efficacy issues or the efficacy question is a little bit secondary, meaning we know that it's not the perfect trial design to measure whether or not it works. And when we expand to the children, we plan on kind of taking it to the next step, not only in decreasing the age requirement, but also, making it a better trial to be able to determine efficacy.
Thank you. There's a question around Kabuki syndrome as an epigenetic condition and some of the education that we did around what that means. There's a question that says, does that mean that the child could have gotten Kabuki syndrome because of something in the environment during the prenatal environment? So does epigenetics play a role? Is that sort of the formation, I suppose, or the origination of the condition? I'm not sure who wants to take that. Maybe Jackie, that's one for you as well.
Allison, do you want to take that one?
Please.
So for prenatal, no, the environment would not have an impact on it. The environment does play a role in epigenetics, but that's kind of like when you're personally exposed to it later on, there could be some effects, but prenatal, there is no effects on environment, and linking that to Kabuki syndrome.
Okay.
So
great.
There's absolutely nothing that anyone could have done differently to make their child not have Kabuki syndrome. Nothing. It was just a random accident that occurred, a random spell check error in the DNA. There's no environmental impact. And when we say epigenetic, we're talking about because KMT2D or KDM6A, their function, even though it's just this one protein that's broken in Kabuki syndrome, their function is to affect the ability of other genes to make proteins, which is the function of genes. And that's why we call them epigenetic disorders. Has nothing to do with the environment. And I understand why that is totally confusing because epigenetics is used differently in other contexts.
Thank you. That's very clear. There are a number of questions that pertain to outcome measures, and I'm going to combine a few questions because there are some about outcome measures with the modified Atkins study and then some around the HOPE study. And a little bit around, how are those going to be analyzed and will they change between adolescents and adults? So maybe, we're talking about a specific battery potentially of tests and sort of trying to build that clinical trial readiness program. This might be an opportunity to talk a little bit about that as well, but kind of open it to the floor to whoever wants to tackle that one.
Well, Jackie, I can certainly speak to the HOPE trial and you can chime in as well. Or if you'd like to start with the Atkins, your battery and app and the Atkins trial, then please do so.
No, go ahead, Michael, because it's relevant to both. So you start.
Okay. So the way we selected the measures, if you go back to Jackie's presentation, is what are the things that are most impacted in the population and how can we measure it? And then we tried to identify the most sensitive, reliable tests that can measure this and that are age appropriate for the different folks involved, because remember, we have adolescents as well as adults. So we picked a number of tests that measure visual, spatial, and motor coordination abilities, as well as visual memory and verbal memory. And in addition to that, mood, and in terms of how that affects your quality of life or functional measurements. So that's the types of tools that we picked here and why we picked the ones we did pick. How we're going to measure this is we're going to look at change over time. So every person is like their own baseline, and then they will come in, at least in HOPE, and I'm sure it's the same for Atkins. It's just what is being modified is different. And they will get a battery of tests before they have anything that they do differently, whether it's changing their diet or taking a drug or placebo. And then we will measure them on these same tests over time. And then at the end, we're going to see how they perform or how their performance is at the end compared to how their performance is at the beginning. Now, when you aggregate this data across all subjects, then you see how the groups did, broken down by whether they received the active treatment or placebo. Does that answer your question, Leila?
Yeah, absolutely. I think it does. And there was a question maybe around, will these be different for different subsets of the population? I know that we're obviously constraining, or we have an age limitation for the HOPE trial. But eventually when things are tested across the population, I guess we will have different, different tests that we will administer, obviously, to different segments of the population.
Right. So as an example, if you're going to measure visual, spatial, motor coordination, and you're going to look at a test that is used both in adults and children, you have something that's called block design. And the block design is found in the Wechsler Adult Intelligence Scale test battery, the WAIS-4. But it's also found in the Wechsler Intelligence Scale for Children, version 5. And it's just how hard is the task for the participants to do is the difference between which version they get, for instance. For other tests in here, we tried to pick tests that are appropriate across the age range. And then where there aren't tests that are as appropriate for across the age range, we tried to modify how these tests are given or administered so that they can be appropriate across the age range. Does that answer your question, Leila?
It does. It does. And Jackie, was there anything you wanted to add from the modified Atkins perspective?
Yeah, so I mean, it's the same idea, right? It's where we're really looking at tests that we can, like Michael said, adapt across. And when we can't, we are looking at tests that are comparable to each other and within the same kind of domain, if you will, as far as measuring the same sort of cognitive function. So we can really get an idea of how to do that across time. And I don't know if Adam wants to talk about, Redden Lab's philosophy on that a little bit as well.
I can talk generally without being specific to Reading Lab. We focus a lot on, lots of clinical trials focus on measures that are functional and are related to quality of life, because you can do a test like that block design one that Michael mentioned, but how does that relate to someone's ability to interact in the classroom or talk to their friends or be able to get a job? Some of those things need to have a direct connection with how people function within real life. And so it is really tricky getting a battery that covers both that impairment level where you're trying to pick up the things that don't work as well, and then also the things that have bearing on success in life. And because we all want to be able to remove some of the things that are prohibiting a successful integration into society. And from our point of view, talking is about as important as it gets.
And actually on that front, maybe back to Dr. Ropacki, is patient reported, the feeling that they are integrating better with their friend group, or that things are easier for them, or maybe parent reported or carer reported? Is that taken into consideration with regards to the FDA's assessment of success?
So the FDA is very much focused on patient-centered drug development these days. They do like to hear from the caregiver their points of view. They like to hear from the patients their point of view. Whether they would allow those endpoints to be used in their decision making as the primary, that has never happened thus far in trials. And I'm not just talking about Kabuki syndrome, I'm talking in general. So there's a bit of a disconnect between the patient-focused drug development mandate and what the Food and Drug Administration actually allows and looks at for trials. But to make sure that we've covered our bases, and Jackie can speak to her modified Atkins trial, but we've included things where the patient's point of view can be included, as well as the caregiver's point of view as well. And then on top of that, performance-based tasks, such as the Redden Labs tasks and other visual, spatial, motor coordination, visual and verbal memory tasks, so that we've captured every type of what's called a clinical outcome assessment. Every flavor of it, if you will, is captured in our battery so that we can do a full 360, including quality of life, which was mentioned earlier. Right.
And given that I'm paraphrasing one of the questions here, but given that we are trying to restore this balance between open and closed chromatin, and exactly what you've described in this session, do we think there will be also downstream benefits in other areas that Kabuki syndrome impacts? So things like muscle tone and immune profile, and maybe hyperinsulinism and other areas that can affect some Kabuki patients, are we measuring that at all as a part of the outcome of the trial?
So we are measuring a number of these things, and we're, again, no pun intended, we are hopeful that they will be modified. What I can say is that as these are not things that are known, as you've alluded to, you cannot put them in a protocol as like your primary thing that you're focusing on, or your secondary. You have to make them more exploratory things that you're focusing on. And that's just because these are things that are questions, they're unknown. So if they're not known, then you explore whether or not you do have an impact on that. And then if we're so lucky to go to the next phase of development, some of those things that were exploratory and you didn't have data on can be elevated up in the food chain, if you will, of your battery of assessments, and you can make them a primary or you can make them a secondary based upon the strength of the evidence and the willingness of the Food and Drug Administration and other regulatory agencies. Jackie.
Okay.
Leila, can I add? Actually, maybe Jackie was going to do this. You're going to talk about immune function, Jackie?
No, you go ahead. I was going to also talk about hypotonia, so go ahead.
Oh, okay. Well, I'm just quoting your data. The mouse model that Dr. Johnson established and that you investigated showed improvements in immune function in terms of IgA. So we are hoping that these will also be carried forward to patients or people with Kabuki syndrome.
And I, because hypotonia was specifically brought up, I wanted to say that, remember that in addition to it being something that we want to change, we also have to be able to measure it. And currently there is no good way to measure hypotonia in a way to use it in a clinical trial. Hypotonia is assessed by me coming into the office and, feeling the patient and saying, yep, hypotonia, right? And the FDA is not going to buy that, and as they shouldn't, right? That's not a thing that we can measure before and after as an outcome measure. And so hypotonia is controlled by the brain. And so we are targeting this to improve brain function, whether it's the modified Atkins diet trial or the HOPE trial. The goal is to improve brain function in Kabuki syndrome, because that is what you all have said over and over and over again is important: cognitive, behavior, anxiety, and hypotonia, right? So we're targeting this to change the brain function, and we're measuring that brain function in the best way we know how, specifically to Kabuki syndrome, with visuospatial measures, behavioral measures, and speech measures.
Fantastic. Well, I think I speak for everyone when I say how hopeful we also are, to use the pun, for what lies ahead. And just a huge thank you to our esteemed panel. We did have a number of questions, and I just wanted to reiterate what Amanda's written in the chat, which is that we are collecting every question, and certainly will, for those we weren't able to go through orally here, we'll be following up after the conference and sending responses to everyone.